Welcome. Happy Monday. We are coming down the home stretch of the semester here. So what I want to do today is first go over the different things that are going to be due, the activities due for lecture, and then continue our discussion of the nephron. As a reminder, attendance in the WebEx live is not mandatory. We have to watch it at some point. And I try to keep them to about 20 to 30 minutes, hopefully around 25, with the idea that you'll spend the rest of the time working on all the stuff that I have assigned for you and actively engaging with the material, whether that is reading the book, taking notes, going back through, listening to the video again, doing the different activities, making flashcards, all of that good stuff. Okay. As always, please feel free to type in the chat any questions that you have. And we're going to switch gears and go over to your Canvas site as just a reminder of what you have coming up this week. Okay, so as a reminder, upcoming this week, so you've got another activity for lab. The pre-lab, remember that pre-lab quiz that you did was for all 3 lab weeks. So 1 pre-lab quiz, 3 lab assignments. This next one is due on the 23rd at midnight. It's very similar to what you did in the first week, so please make sure I graded all of that and I gave you guys comments on what you messed up on. So if you made some mistakes, please make sure you look at the comments and you look at what you did wrong and improve upon it. Otherwise, you'll just keep getting penalized because it's very similar. Due the 24th at midnight is for you to draw me a little diagram of the renal aldosterone angiotensin system, and we're going to go over that more Wednesday. So I would stay tuned on that one. You're going to draw it for me. You're going to take a picture. You're going to upload that picture. Then next Monday, you have your paper due. Everyone should have received comments for that as well on your draft. If you submitted a draft, the final is due. Please make sure you read the assignment carefully and make sure you're meeting all of those criteria. And Dr. Pogue is grading the first half alphabetically. I'm grading the second half alphabetically. You should know that based on who gave you comments on the draft. But if you have questions, please email one of us and we'll direct you to whoever is grading your assignment. Okay. The other thing due on Monday is the Unit 4 questions to discuss. So if you look at your Unit 4 questions to discuss, there are 8 questions. This will take you a little while. I would keep a running Word document, and as we get to them, answer them. We have gone over a few of them at different points. So you can start working on at least some of these. Just throw it out there, get it done now as opposed to waiting till the last minute. So that's what we got going on this week. Questions, I'll take them in the chat. Otherwise, I'm going to switch gears here and go to the PowerPoint. Okay. So where we had left off is we are breaking down the nephron bit by bit, segment by segment, and talking about what each segment does, what the role is. And we're keeping in mind the goals of the kidney, the things that the kidney has to get done, these ideas of ion and water balance, about blood pressure, about regulation, all of that we need to keep in mind when we look at the different parts and see what aspect of each segment of the nephron contribute to those different goals that we have. Okay, so we had left off talking about the loop of Henle, how it's this sequential uptake of water in the descending loop of Henle, and then salt in the ascending loop of Henle. And that is, again, based on both the transport capacity, so what proteins are in those membranes that allow for the movement of different substances, as well as the gradient. So in the descending loop, this is where your aquaporins are. Aquaporins, channels for water. Water is going to flow. In the ascending, there are no aquaporins. Water can't move. So instead, salt is going to move. And again, there is this gradient that's formed. And as the osmolarity decreases, salt is able to move through things like the NKC cotransporter. And the size of the loop will determine how much water is able to be reabsorbed. Okay. This is showing you that osmolarity gradient. So up near the proximal and distal tubule, it is the same osmolarity. It's isoosmotic to blood, so around 300 milliosmoles. However, as you move deeper into the loop of Henle, you're going to see that that osmolarity increases. We talked about this. This is where we left off last time, that the descending, again, is going to move water through aquaporins. Ascending is going to move salts. So it's going to maintain a gradient through the use of the sodium-potassium ATPase, which will keep the concentration of sodium low inside those epithelial cells and will allow sodium to then fall down its concentration gradient to go into the cell, bringing with it potassium and 2 chlorides in that NKC cotransporter. Okay. Chloride can then flow down its gradient and enter that peritubular fluid, and potassium can also go back into the lumen secretion. Okay. The distal tubule is where we're going to see the majority of our potassium secretion and our sodium and chloride reabsorption. And this is also where hormones can act for water recovery. So the distal tubule, hormones that we'll talk about more on Wednesday, things like ADH and PTH, they act here. Okay. And the distal tubule is the site where we reabsorb most of the remaining sodium and chloride through our epithelial sodium channel, or ENaC. Okay. So again, this is showing you a picture of our distal tubule with the lumen here and the peritubular fluid that will lead into the blood vessels. And so we're going to use different channels to help build that gradient. Again, gradient driven by the sodium-potassium ATPase. And we're going to move the majority of our sodium and chloride here into that peritubular space. Now, the interesting thing about the distal tubule are the aquaporins. The interesting thing about the distal tubule are the aquaporins, because the expression level, the amount of aquaporins that are expressed on these membranes can change and can be upregulated in times of dehydration. So as we'll talk about with hormones, some hormones will increase the amount of aquaporins here, increase the amount of water that can be reabsorbed. This is also the main site for potassium secretion, so that sodium-potassium ATPase is also going to drive a large concentration of potassium into the epithelial cell, which will then fall down its gradient and enter back into that filtrate. So secretion, remember, is going from the blood vessel into the filtrate. And this distal tubule will eventually lead to our collecting duct. And our collecting duct is the spot where we have ion and water regulation again. And here we're talking about two different kinds of cells. We have principal cells. These are cells that secrete potassium and reabsorb sodium. And we have intercalated cells that are involved with pH balance through protons and bicarb. And the principal cells can respond specifically to our potassium need as well. So the principal cells can be stimulated to secrete more potassium or less potassium depending on what the body needs. And intercalated cells in the same way can be stimulated depending on what the body needs in terms of potassium balance. Okay. So that is the overview of the segments, what they briefly do. Now let's dive into a few key concepts. So we went over briefly what each segment does. Let's talk about a few overarching concepts. And the first is that of the countercurrent multiplier. So that we say that the loop of Henle acts as a countercurrent multiplier. What this means is that it's going to have fluid moving in opposite directions to maintain that gradient. Fluid is going to move first down and then up to maintain that gradient. We have maintained the gradient of low osmolarity at the top near the cortex, high osmolarity deep in the medulla. And this is built and maintained by the loop of Henle structure, as well as by the transport of solutes and water in the collecting duct and the interaction it has with the capillary system of the vasa recta, which is the name we give again for some of those capillaries that are around the loop of Henle. Keep in mind that the descending, only water can move. Now, you might think that water moving out of the filtrate in the descending loop of Henle would destroy the high osmolarity, right? Because It's got a lot of salt down there. Lots of water is going to want to leave and decrease that saltiness because of that high osmolarity, right? Water will follow that salt. But it's balanced by the ascending loop pumping more salt into that fluid. So the descending loop is going to kind of water it down, but the gradient will be maintained by the transporters on that ascending loop. And this is what that looks like. So if there was no osmotic gradient, then the active transport of sodium chloride and potassium would start, and it would start to increase the osmolarity. And then water would then move into that higher level of osmolarity. And we would again reach that isoosmotic state, okay? But because we continually have more transport of salt, we're going to keep building that gradient. And water will keep moving, but we'll keep putting more salts. And eventually, we'll get this gradient that's formed because of the balance between salts coming in, balancing out what the water does. And this process will continue until we get a full osmotic gradient established. So we said that the collecting duct can help form the osmotic gradient. And this is where the waste product of urea comes into play as well. Now, you might remember when we showed that chart about how much urea is reabsorbed versus filtered, and there is actually a fair amount of urea that doesn't end up in urine, which is interesting because urea is a waste product, and one of the goals of the kidney is to get rid of waste. But in fact, the kidney actually uses some of that urea to help maintain the saltiness in our osmotic gradient. So you're not going to get rid of all of your urea. Instead, you're going to reabsorb some of it, or you're going to reabsorb some of it so that you can maintain that osmotic gradient. It's going to add to that saltiness. And this happens in our collecting duct. So the collecting duct is the spot that is permeable to urea. So as water leaves the urea concentration, as we move through our epithelial tissue, starting again in that proximal tubule, down the descending loop of Henle, up the ascending loop of Henle, one of the things that's been happening is we've been concentrating our urine, reabsorbing more and more water. And our concentration of urea has been increasing and increasing and increasing in this primary urine, or this filtrate. Deep in the medulla, the collecting duct will now express urea transporters. And this will allow urea to go into the interstitial fluid and increase the osmolarity of our medulla. So not only are we increasing osmolarity through the transport of salt, we're also increasing it through the transport of urea. And this is what that is shown. So we have these specific transporters for urea that are going to allow it to enter, to leave the collecting duct and enter the fluid here, making this super, super salty. Okay. The other component for maintaining our osmotic gradient, I said, was the vasa recta. This is another spot where we have a countercurrent exchanger. For most tissues, capillaries are going to drain the interstitium into the blood. And if the capillaries flowed just from the cortex down into the medulla and out, then it would draw all of the fluids and the solutes out of the kidney, and there'd be no gradient. It moved in just one direction. However, like the loop of Henle goes down and then goes back up, the vasa recta does the same thing to keep the saltiness. So as it moves in, it's going to be at the interstitial fluid is going to be at high osmolarity, and so the capillaries can pick up salts and lose water, right? Salts are at a high gradient, so they're going to move into the capillaries. As it leaves the medulla, it's at a lower osmolarity, so it can lose some of those salts and pick up water, and it will then leave the kidney when the fluid is isoosmotic to blood. It'll leave the kidney when those are both at 300 milliosmoles. That way, there is no net flow of salt or water as it leaves. This is what it looks like. So right now, what this is showing is not the loop of Henle, but our vasa recta, our blood flow. So it's going to enter the nephron at 300 milliosmoles, and as it gets into this osmotic gradient, we're going to have the net movement. Water is going to move, solutes are going to move. As you go deeper and deeper, more and more solutes are going to move in because they're going to move from high concentration to low concentration. As they move out, more and more water is going to flow in, and it's going to end at roughly the same osmolarity as it entered. If it just went in one direction, all of the salt, if it exited down here in the medulla, it would take with it all of the salt. Does everyone see that? How it would take with it all of the salt? Because all the salt would want to move from that high concentration into the lower concentration of the blood, and it would exit. The gradient would be destroyed. Chaos in the nephron world. But instead, it does the same thing the loop of Henle does. It goes down and then it goes up. And in this way, it helps to maintain that osmotic gradient. So our nephron has these key structures, this key setup, this countercurrent multiplier that's down and then up to help maintain that osmotic gradient, because the osmotic gradient is what drives the reabsorption of water. Okay? This is how we concentrate our urine, making this super salty and keeping it super salty. Okay. This is the same thing shown again. Osmolarity as the filtrate changes. First we move water, then we move salt, and at the very end, we move urea. Let's talk about how these solutes move specifically. In general, solutes are going to move by secondary active transport. Remember, active transport uses ATP directly. Secondary active transport uses the gradient of something else. So these cells are going to use that basolateral sodium-potassium ATPase to make a sodium gradient, to keep the concentration in the epithelial cell of sodium low, so that sodium will want to move into the epithelial cells, and with it, it'll take some buddies, right? So in the proximal convoluted tubule at the very beginning, things like amino acids and glucose are going to use transporters driven by that sodium gradient, right? Our old friend, the SGLT. Like, come along with me, glucose. Follow me. I have the gradient. I have the power. In the loop of Henle, we talked about the sodium potassium chloride cotransporter, where potassium and chloride tag along with sodium's gradient. And in the distal convoluted tubule, calcium does the same thing. We got a calcium antiporter. So that's how calcium is able to move, is again via that sodium gradient. Okay. I know that was a lot of information. A lot of information about renal functions. If you want to go back, this would be a great time at the end of this class. If you're live in the class, if you're watching the video, pause it, read through these pages in chapter 10, 507 to 514, 519 to 521. Okay. We're now going to briefly talk about, I'm going to start talking about the regulation of renal function. And this is what we've been working on in the lab. So we've been working on glomerular filtration rate, GFR. So what we're going to do is we're going to talk about that again, and then explain what affects GFR. And then we'll talk about some key hormones that affect kidney function. And we'll end by talking about what the hypothalamus does and how the hypothalamus can regulate this whole thing too. Okay, so endocrine hormones help regulate ionic and osmotic balance. They act on both the cardiovascular system and the kidney to do this. But the time frame here matters. So our mineral corticoids, which we'll talk about, act over hours because they're affecting transcription. So when you think about time frame, you want to think, what is this hormone doing? If it's got to go to the nucleus and turn on genes, and the genes have got, the mRNA is going to be made into protein, the protein's going to get to the right spot, that's a lot of time. We're talking a long time for that to happen. Whereas peptide hormones, I can just like bind to something on the membrane, start a cascade, they're going to be rapid, okay? So we'll talk about those two different types of hormones, and some vocab you need to keep in mind. Diuretics mean you are going to stimulate urine production, lots of excretion of water. Diuretics, excrete your water, you're going to pee a lot. Antidiuretics, you're not going to pee. You reduce the excretion of water. So GFR is affected by hydrostatic and oncotic pressures. So filtration across the glomerulus is affected by the surface area that's available. So how much space is there for things to move from the blood into Bowman's capsule, as well as the pressure gradient. So how much is there a push across the filter coming from our blood pressure? So is there a lot of blood pressure pushing things across the filter? And then how much space is there in the filter for things to move through? So GFR, again, has to do with the volume of plasma per minute that it takes for something to get into the filtrate. And it's driven by the glomerular capillary hydrostatic pressure. That's the hydrostatic pressure coming from the capillaries, the hydrostatic pressure in Bowman's capsule, and then the net oncotic pressure, which we'll talk about. We'll define all these fun terms. Okay, so again, we're talking about the movement of plasma from the glomerulus to Bowman's capsule. For the average human, your GFR is going to be around 125 milliliters a minute, or around 180 liters a day. And the filtrate has to cross 3 barriers to enter Bowman's capsule. It's got to go through the endothelial layer of the capillary. It's then going to go through the endothelial layer that surrounds, and then in between, there's a basement membrane. So it's got to pass through 3 different filters, if you would. Now, your textbook has this metaphor, and I don't know if this is helpful for you. I don't know how much gardening you do. If it's not helpful, leave it. If it is helpful, take it along. So your textbook talks about GFR in relation to like a garden hose. So if you imagine there's a garden hose along your house, and midway down the hose, there's an area that's got a few small holes in it, okay? We can call the tap that is on the side of your house, the garden hose that's attached to that. Imagine that's like your heart. And then the area with the small holes, that's the glomerulus. The first section of the hose before you get to the small holes, that's like our renal artery. And the section of the hose after the small holes, that's like our efferent arterial. So we've got the afferent arterial, the efferent arterial, the glomerulus. Blood coming in, filtering, blood coming out. If you start to close the tap a little bit, you're going to have less water leaking out. So if you decrease your cardiac output, then there's going to be less filtration rate, less stuff passing from the plasma through the filter per minute. Okay, so again, you decrease, turn the knob on your house, decrease the amount of water flowing the tap is like decreasing your cardiac output. And that's going to cause less water to trickle out those holes in your hose because less water is passing from the beginning. So your GFR is going to go down if your cardiac output goes down. Now, if you take a look at the hose and you squeeze the first part before anything reaches the part of your hose that's all messed up, you block it upstream of the holes, less water is going to go out, right? That makes sense. Squeeze the hose, nothing is passing through. In the same way, if you lower the blood pressure, the pressure going in, you're going to lower your GFR. If you block downstream of those holes, however, if you squeeze after the holes, you're going to shoot more water up. This is what happens with vasoconstriction. So if you squeeze the arterial afterwards, you're going to get more up in the GFR. And so these are some ways that you can modulate filtration rate depending on these cardiovascular principles. With that in mind, we also have to talk about how there is pressure. There's not just pressure from your arterial system going into the filter. There's also pressure on the other side. The glomerulus does not empty into a vacuum. It does not empty into just a blank basin or empty into space, right? It's opposed by the fluid. There's its own hydrostatic pressure. So the interstitial fluid's hydrostatic pressure is around 15 millimeters of mercury, which is a unit for pressure. And the glomerular capillaries are pushing on it with about 60 millimeters of mercury, which creates a driving force of 45. So it's pushing with 60, it's opposed by 15. This creates a driving force of about 45. However, moving proteins can't pass through that filter, and they create their own kind of pressure because they hit the filter and they bounce back and they can't get through, and so they remain in the blood. And we call this the oncotic pressure. So it's the osmotic pressure that comes from that protein concentration gradient, because there is going to be a lot of proteins in the blood still, not a lot or none in the filtrate. That has its own pressure associated with that gradient, and that is an opposing pressure of around 30 millimeters of mercury. So this brings our net force to 15. So we take the pressure pushing on it, 60, being opposed by the hydrostatic pressure in the interstitial fluid of 15, and being opposed by that protein gradient of around 30. So it brings it to 15. And this is what that looks like. So again, this is our afferent, our approaching arterial, our exiting arterial. If we zoom in here, we've got blood pressure pushing with 60 millimeters of mercury. That's our hydrostatic pressure gradient. It's opposed by the hydrostatic pressure in the lumen, the filtrate itself of 15, and it's opposed by the oncotic pressure of that gradient of all the proteins being in the blood, none of them being allowed to be in that interstitial space. Okay, so 60 minus 15 minus the 30. Now, we also have, this is just talking about the forces favoring filtration. So again, 60 is our glomerular capillary hydrostatic pressure, and it's usually high because of the resistance in that afferent arteriole. It's like squeezing the hose after we poked all the holes in. Now, if it's low, Bowman's capsule oncotic pressure can be brought down low if there's a change in the protein gradient. Now, if we change the hydrostatic pressure, if we change our blood pressure, we can change all these numbers. Okay. Showing you all that math. So if we change the blood pressure, we can change our filtration pressure. Okay. So things to think about before next class. I want you to think about how we can maintain a constant GFR as blood pressure changes. So as our blood pressure is going to change, we're going to change the driving force across the filter. And what that could do is it could change the amount of stuff passing into our filtrate. But our body doesn't like that. The nephron wants to maintain a constant GFR. And so keeping in mind that metaphor of the house, of the garden hose, and the holes in the middle, think about what kinds of things your body could do to maintain a constant GFR as the pressure in that afferent arteriole changes. Okay. So that is where we're going to stop today. We talked about the rest of the segments of the nephron, and then we started to talk about how we regulate nephron or renal physiology. We'll pick up on Wednesday by talking about intrinsic versus extrinsic factors, things that are made by the kidney versus things that are made in other parts of the body that affect the kidney. We talked about those brief things you have to do. That is going to do it for today. If you guys have any questions,